Thanks, everybody. Today we've got Dr. Kim Connelly from Toronto, who's going to be talking to us on some very exciting novel imaging techniques in his field, which is quite interesting. He studies a whole bunch of things to do with disease in the preclinical era of rodent imaging, which is something that I've never done. And so he looks at models of cardiac disease, especially in the field of diabetes and cardiac function, and is really quite a renowned young investigator from across Canada. He's won multiple Young Investigator Awards and currently has a lab at the Keenan Research Center at St. Mike's. So it's really a pleasure to have you talk to us today, and I think you're going to be talking about some stuff that a lot of people on the line won't know a lot about, which is great, which is hyperpolarized C13 imaging, which is something that isn't done a lot of, and only a few centers in Canada have the capacity to do it, and yours is one of them. And so we're very interested in hearing what you're up to and how we can use C13 to understand disease pathophysiology. So thanks a lot, Kim. We really appreciate giving this talk, and take it away.
No problem. Okay, thanks, James. Thanks for the kind introduction. So what I thought I would do today was to give you a little background into metabolism and hyperpolarized carbon-13 imaging. And really the backdrop to this is hopefully in the next few weeks we should be doing the first-in-man studies. So this it won't be the first time this technique's been used in man. It's actually been done at UCSF in California, where they injected hyperpolarized pyruvate to look at prostate cancer, but this would be the first in man cardiac application. So there's a fair bit of background, because otherwise I think I'll lose a few people, but I'll fairly quickly skip through that and then get through to the exciting stuff. So this is a nice quote I found from Lynn Margulis, and if you don't know her, she was married to Carl Sagan and is really sort of attributed as being probably the best biologist and evolutionary biologist that the world's ever seen. A very insightful woman, and she noticed that the body continuously self-repairs. Each 5 days you get a new stomach lining, you get a new liver every 2 months. Your skin replaces itself every 6 weeks. 98% of the atoms of the body are replaced each year. And this is a non-stop chemical replacement process, really encompassed by the overarching term called metabolism. So clearly metabolism is an important aspect, but as you'll see, it's difficult to study, and that's limited our understanding of it. Now, it is an MRI talk, so I'll give the standard slide that hopefully everyone is already a convert to MRI. But the real thing I want to focus on here is typically when we do MR imaging, we look at proton imaging. So we're looking at the image obtained by the signal from hydrogen. And as you know, the reason that this is able to be done is because the body's about 70% water. So even though the actual signal from each individual proton is small, because the concentration in vivo is approximately 110 molar, because of that fact, we're able to actually generate a signal. And clearly, MRI still remains the gold standard in the assessment of cardiac function. And then over the last 10 to 15 years, there's certainly been an explosion of data to demonstrate that it is a wonderful technique to look at tissue characterization. And a lot of this has been led by Canadians looking at T1 mapping, T2 mapping, T2 star mapping, and of course the workhorse tool, which is late gadolinium enhancement. And obviously this has been very useful in revealing a significant amount of information overall in terms of fibrosis, viability, and tissue composition. And as MRI has progressed, really the issues that we see from a cardiovascular viewpoint have changed. So I want to just switch gears now and just give you some few insights into to heart failure, because I think this is really where we want to take this hyperpolarized carbon-13 imaging technique. And why is it so important? Well, if you have a look at the numbers now, heart failure in Canada is still a very major problem. So about 5,000 deaths a year, 100,000 hospitalizations, 20% readmission rate, estimated costs somewhere in the order of billions of dollars per year. And you're probably thinking, well, you know, as MRI's gotten better, we can make better diagnoses, we have better therapies. Well, this is data from the Toronto group looking at the actual incidence and outcomes of heart failure. This is Ontario data from 1997 to 2007. And what I want to bring your attention to here, if we look at the overall mortality rate in 1997 and compare that to 2007, if you look at the 1-year mortality rate, 25.9%, when it's risk adjusted 27%. So that means if you turn up to a hospital in Ontario with a diagnosis of heart failure, fairly simple diagnosis to make actually, you'll be 1 in 4 will be dead. Now if we fast forward 10 years and look at what the outcome has been during that time. Now during that time, remember, we've more frequently used advanced imaging modalities, we have much better therapy, we have better interventions, more people are getting PCI, coronary artery bypass grafting, automatic implantable cardiodefibrillators. We've seen an absolute change of -6%. So that's a relative reduction of -1.8% absolute change. So yeah, we've reduced the outcomes, but still basically 1 in 4 people will die because of heart failure as contemporarily as 2007. I can tell you the more recent updates of this show, the numbers are roughly the same. So yeah, we're making inroads, but we certainly haven't made as substantial as inroads as we would like. And I think one of the reasons for this is there's a lot more than to meet the eye when we look at the heart and the outcomes with heart failure. It's not just a matter of focusing on the coronary vasculature. We've got to continue to go back and look at the substrate and look at the myocardium. And that's really where metabolism comes in. So I think if we go back as far back as 1939, really some of the older gurus who looked at metabolism, they first hypothesized that there was a starvation hypothesis. And what they were really saying there was that there's just not enough energy to maintain the heart to beat adequately. And since then, there's been a number of other studies using P31 spectroscopy, positron emission tomography, as well as in vivo and in vitro biochemical studies, which have gone on to demonstrate that one of the fundamental problems in the failing heart is that there is an alteration in how the heart metabolizes and also what is available for the heart to metabolize. And I put in this nice schema here. You can see this is P31 spectroscopy data from Stefan Neubauer's group, demonstrating that if your phosphocreatine ATP ratio is using a 1.6 cutoff, that in patients who had heart failure, you saw a substantial change in overall mortality. Again, just providing evidence that in the failing heart, metabolism is a key feature. And one of the other things that's very important when we look at heart failure overall, so I've told you heart failure is bad, 25% death in 1 year. Now, it doesn't matter whether or not you have a normal ability of the heart to pump, that is a preserved ejection fraction, or a reduced ejection fraction, the overall mortality is the same. And the reason that this is very interesting is if we look at patients who have a reduction in their heart function, of those patients, there are multiple evidence-based therapies. We have at least 4 therapies which have a clear mortality advantage. If we look at that group of patients which have preserved ejection fraction, currently there are 0 evidence-based therapies. This makes up about half of those patients who present to hospital. So in half that group, it's probably the biggest cause of healthcare expenditure in, at least in Ontario, expected to make up to $1 trillion in the US by 2020. We have 0 therapies to treat that group. So clearly this is a huge problem. So anything we can do to better understand what's going on in the heart failure is going to be important.
Calcium elevated around the myofibrils.
Now,
myosin binding sites on
let me just turn that off. So what about the heart? Let's just get into a little bit of detail about the heart itself. So as I've said, everything about the heart is about contraction and relaxation, and this is an active process. So if you think about the average weight of a heart, it's about 250, 350 grams. It must produce approximately 6 kilograms of ATP every day just to function normally. So 35 times its own weight. So that's a pretty extraordinary figure. And remember, as soon as you exercise, as soon as you do anything else, it's got to generate more ATP. Now, in order to do this, it will actually use basically anything. Fatty acids, glucose, lactate, amino acids, anything it can. Because clearly if the heart stops pumping, you're in a little bit of trouble there. And one of the things that a lot of people fail to remember is if you actually remember how the heart contracts, and this is probably best described by what's called the Raman hypothesis, where you have a thick myosin filament and a thin actin filament here. It all happens in diastole, and diastole is actually the Greek for the heart to relax. And that's when you generate ATP to move this so that it's in a position to enable the power stroke to occur. And it's also required to use all those ion pumps which maintain cardiac homeostasis. So although we focus a lot on systole, it actually all happens in diastole. But just remember, 6 kilos of heart every day for each of our hearts to generate 6 kilos of ATP. And obviously, in order to generate that, this is a very complicated process. And typically, we break it down into 3 integrated components. Substrate utilization, that is taking things like fat that you eat, glucose in the diet, and converting that into a form where it can undergo oxidative phosphorylation, by which then you can generate ATP. Once you've generated the ATP, then you've got to transport it back to the location where it needs to be used. So you need to think about this in these nice 3 separate but very integrated components. So it's worthwhile just doing a brief overview just to give you an idea of how the heart works. And I don't want to bore you too much, but it will make a lot of sense when I show you the data of what we're doing with the C13 later. Otherwise, it really is not in context to understand why C13 is probably the key imaging technique to look at metabolism in the heart. So this is a compartmentalized figure of a normal heart. And what we have here, this is the blood, this is a cell, and here we have the cytosol, and here we have the mitochondria. And most energy, or ATP in the heart, is produced by this process known as beta oxidation. That is, you eat something, it gets metabolized, turned into a fatty acid. There's a transporter on the membrane of the cytosol which flips it into the cytosol. It then is converted into a form where it can enter the mitochondrial, and really it's converted into this particular moiety here known as acetyl-CoA, which can enter the tricyclic, or the Krebs or TCA cycle, and from that and during the electron transport chain, we can then generate ATP. So the importance of fatty acid oxidation, about 70% of ATP is generated. It provides the greatest amount of ATP generated per unit of substrate consumed. So it's the best way to get ATP, but there's always a cost with anything, and the cost is it consumes more oxygen. Now, why is this important? Well, in a state where you don't have extra oxygen available, such as in the ischemic heart, this tends to generate oxygen-free radicals, which uncouples the entire oxidative phosphorylated process. So you can see immediately why ischemia is a problem in terms of generating ATP. Now, importantly, there are a number of committed phases, and I really just bring this up to demonstrate that these are intense areas of research for the drug discovery, because potentially if we can modify these in the disease state, we may be able to then modify how the heart generates ATP, and this may be a beneficial process. Now, what's the other way in which the heart generates ATP? Well, this is the one that probably everyone's better familiar with. This is glycolysis. So this is when glucose is metabolized ultimately into glyceraldehyde 3-phosphate, and then undergoes a number of transformation steps into this substrate here called pyruvate. Once its pyruvate's generated, and importantly, to get from glucose to pyruvate, this actually consumes energy. So you lose ATP getting from here to here. But then, once you've generated that glucose, it can then enter the mitochondria and be generated through acetyl-CoA and generate ATP again, or it can undergo a number of other processes. And the other one that you'd obviously know is in the anaerobic oxygen-free environment, when you need ATP quickly, what it does is it's rapidly converted to lactate. So that's basically the key ways in which the heart generates those ATP: beta oxidation or glycolysis, and then from glycolysis you can undergo decarboxylation to acetyl-CoA and enter the TCA cycle, conversion to lactate, or this other pathway which we won't go into called carboxylation or anaplerosis, which is a way to cycle nitrogen throughout the heart. And again, just like beta oxidation, glucose metabolism, glycolysis is heavily regulated. But well described many, many years ago was by this gentleman Randall, was this hypothesis that there's a reciprocal relationship between the two. That as you increase your ability to undergo beta oxidation, you dampen down your ability to undergo glucose metabolism or glycolysis and vice versa. And this is thought to exist because obviously in each of the steps you consume a number of other moieties that are involved in the reaction, and it's designed so that you don't completely deplete one aspect and then completely turn off your ability to undergo beta oxidation at all. So that's really a bit of a very rapid walkthrough in our understanding of glucose metabolism. Obviously, if you go to our Edmonton colleagues, Jason Dyke and Gary Lopez-Chucks, they really are some of the world experts. So hopefully I won't insult them if they ever get onto CMR and listen to this talk. But you know, it's a very complicated process. But this is really the key components, and I'll show you why this is important currently. So what happens in a disease setting? And I'll just give you two examples here. And the two best ones are what happens in ischemic heart disease and what happens in the presence of diabetes. Well, I mentioned before, obviously in the normal heart, predominantly 60 to 70% free fatty acid and about 30% glucose metabolism. Well, what happens in diabetes, of course, is it's not a matter of the heart actually running out of fuel. It's a matter of that there is so many free fatty acids running around and so much glucose in the system that the cardiomyocyte actually becomes overwhelmed, overloaded with substrate, and it doesn't know what to do with them. So this transporter here, a non-esterified fatty acids, is not an active transporter. So if you have too much non-esterified fatty acids, they will enter the cell, and the cell's got to do something with them. So what does it tend to do? It tries to undergo as much beta oxidation because it wants to normalize the process. Remember I said before, through the reciprocal agreement with the Randall hypothesis, as you increase this, you actually inhibit the ability to undergo glycolysis. However, as you know, with diabetes, one of the things that happen is you have extra glucose. That is also transported into the cell, and that means you have increased glucose intermediates in the cell. So you lose your ability to undergo glycolysis, you deplete many of the things that you require for beta oxidation, and you generate all these glucose intermediates, and they then undergo downstream inflammatory pathways, fibrotic pathways are activated, and that's one of the key reasons why we think diabetes itself impairs cardiac function. One of the other key features here is that the way in which we believe that the increase in fatty acids inhibit glucose oxidation is this is a key enzyme that metabolizes pyruvate called pyruvate dehydrogenase, and that is directly inhibited. So inhibition of pyruvate dehydrogenase is a key step. What about ischemia? Ischemia is a little bit different in that basically what we see here is, remember I mentioned before, fatty acid oxidation, great way to generate ATP, but the cost is it requires more oxygen per mole of substrate utilized. And if you don't have that, it generates ATP, which uncouples oxidative phosphorylation, which causes a reduction in overall ATP generation. So when in the ischemic setting, what tends to happen is the heart will initially try to compensate. It will increase fatty acid oxidation, but what that will do will generate a whole lot of lactate and hydrogen ions, which then overall impair the ability of the heart again to generate ATP. This then has subsequent effects on these pumps, which then directly impair diastolic and systolic function. So you can see it's all interrelated, but the way in which you get there is very different depending on the underlying etiology. However, as you know, what's the biggest problem with diabetes? Well, ischemia and coronary artery disease. So you can see the two are not mutually exclusive. They can coexist. And that's one of the reasons we do think that diabetes tend to have greater remodeling, for instance, after they undergo a myocardial infarction. So overall, if we think about what happens across a time-dependent fashion, we can see in the normal heart compared to the failing heart that there are grave changes in the way in which we undergo substrate utilization, oxidative phosphorylation, and that ATP transport shuttling as you go from the normal through to the failing heart. But the difficulty we have currently, and one of the reasons we don't have good therapeutic interventions, is we tend to pick up the patients here, right when they've got overt heart failure, when it's probably too late. What we need to do is generate a technique or have the ability using biomarkers to start to measure these things early on and say, okay, well, at this particular point in time, we know we need to focus on fatty acid utilization. At this point in time, we need to improve glucose utilization. We don't have that at the moment, and most of the clinical trials have just given drugs right at the end stage of the disease. So it's not surprising we've had a few hits and a few misses in terms of our therapeutic potential to improve this. And here's the sort of current drugs that we've got out there: perhexylene, trimetazidine, and atomoxamia, which are not really available in Canada, fairly weak data, so we don't really have a lot at the moment to try and improve these metabolic defects, which I think really the way the literature is going, these are really now key to many of the ongoing remodeling process we see in the failing heart. So that's really where hyperpolarized carbon-13 MRI starts to step in as a technique that will enable us to directly measure these biochemical reactions. Now, to give a little bit of historical perspective, as you know, when MRI first came about, really in the 40s and 50s, actually a little bit earlier, when the Nobel Prizes were originally given out, it was really done as a technique to look at structure. It was done because they'd recognized that if you delivered energy, that different structures would resonate at a different frequency, and if we could decode that, we could then understand what the chemical structures were. And as I mentioned very early on, the reason that proton imaging, water imaging, has become the predominant technique is the body has a lot of water in it, so it gives a signal. However, there are many other MR active nuclei in the body. Now, what is an MRI active nuclei? It's something that has an unequal number of protons and neutrons. And what that means is when you deliver any energy, some will line up in one direction, others will line up in the other direction in the magnetic field, and the difference between that is what we describe as polarization. But if you have something that exists in nature that is at extremely low abundance, like C13, because it has an unequal number of protons and neutrons, that yes, you can send in a radio frequency pulse and energize it, but the actual signal you'll receive will be so low it will be lost in the noise. So we can't generate any signal. So remember I mentioned before that the millimolar concentration in vivo of water is 110 molar. The equivalent of C13 in the human body would be 0.0005%. So yes, it's MR active, but no, we can't actually generate a useful signal because as this figure demonstrates here, there is just too much noise. So two things moved forward to enable us to start to be able to understand these metabolic substrates. The first one was this technique known as dynamic nuclear polarization. And essentially what this is, fancy technique where you take a substrate, and the one we're going to focus on here is pyruvate, because remember I mentioned from the metabolic process, pyruvate was key for both glucose metabolism and Krebs cycle dynamics. It's small, it has two carbon atoms that can undergo, that you can put C13 into, so it's very useful that it can undergo this process. And what we essentially do is we take this substrate here, we apply a magnetic field, it's usually about 3.35 Tesla, we cool it down to 1 degrees Kelvin, and it's irradiated with a microwave. And what that essentially does is if you dope that solution with something that has oxygen, that has free radicals in it, you can then transfer the energy in those electron shell into the nucleus of the C13, that MR active nuclei. In essence, what does it do? It means you got more spins misaligning, which means you've got a greater signal. And from that, we can increase the signal approximately somewhere between 20,000-fold, 50,000-fold. And what it also enables us to do is we can do that in approximately 50% of those C13 labeled nuclei that we include in the solution. So this technique is very useful. But remember what I said, we have to cool it down to 1 degrees Kelvin. So most people don't like it if you inject them with a 1 degrees Kelvin solution. That's about minus 272 degrees centigrade. So remember, absolute zero is minus 274. So it's fairly cold. So you have to warm it up quickly. And there's this dissolution technique, and I'll just give you a brief bit of detail on that, which means we can warm it up with very rapidly, and you've got about 60 seconds where you maintain that hyperpolarization signal, so you can then image that substrate within 60 seconds once it's warmed up and injected. So complicated technique. I've automatically demonstrated that there's one problem with it, that is we've got a very narrow imaging window, but what it does is it gives us a very good signal to noise ratio. And this is actually what the machine looks like. This is the SPID lab. which essentially you've taken your substrate like pyruvate, we've popped it in there. You can see these are the various frequencies, 3.3 Tesla, 1.3 Kelvin. We've added in various electrons so that we can undergo the DNP process, and we've irradiated it, or sorry, we've microwaved it to transfer those spins into this MR active nuclei. It takes about 30 to 60 minutes for a standard biological sample that we'd inject into a pig or a human. So it's not a 5-second process. It does take some time. Then, as I said, you'll lose your signal within about 60 seconds. This is the dissolution method. So you rapidly dissolve it in a hot solution. It's up to 7.4 pH, so it is a physiological solution. Decay time's about 60 seconds. And overall, the solution you inject into a human is going to be around 5 to 7 ml. So that's the sort of amount we need to inject. In the pigs, we inject about 5 mils. And again, there's two reasons that pyruvate is the key substrate that we like to do this process of hyperpolarization. Pyruvate's small. That's an advantage. It means we're much more likely to get a stable hyperpolarization process. The other thing is when pyruvate undergoes its metabolic process, it can be converted by alanine or alanine aminotransaminase to alanine. It can undergo conversion to lactate through lactate dehydrogenase, or it can be generated into acetyl-CoA, which can then undergo Krebs cycle. So if you add a C13 into the first carbon position, this will actually be cleaved off into bicarbonate. So therefore we can measure bicarbonate and look at the activity of this enzyme here, pyruvate dehydrogenase. That gives us an insight into glucose metabolism into glycolysis. If we add a second carbon, so at the second carbon position, we put a MR active nuclei, C13 nuclei, unlike in this process here where you generate acetyl-CoA, whereas it's the bicarbonate moiety that has the C13, you can see if at the second position when it's cleaved off, it actually generates acetyl-CoA itself, which will have a C13 atom in it, which we can then measure. So that then enters the Krebs cycle. So therefore we can differentiate what's happening between the Krebs cycle and what's happening between glucose. And we'll only generate a signal from the substrate that we've introduced that's been hyperpolarized. So therefore we won't be looking at the native signal. So we can break down what's happening between what we've injected as opposed to the native substrate that your body, because your body obviously during this process is continuing to metabolize acetyl-CoA. So that's really the reason why we like this, because it enables us to give us a real-time insight into what's going on with Krebs cycle dynamics and glucose metabolism. And pyruvate is very safe. It's naturally occurring. If you inject it, you get a small increase in heart rate. That's the only known side effect of it. It is taken up via a non-active transporter mechanism. So it's a very nice marker to use. Now, just two other comments here. With pyruvate, a lot of people ask, well, what's the difference between what you're doing and what, say, you might do in PET imaging? So the most common thing that we would use in PET imaging would be 18-FDG. Now, for 18-FDG to get into its cell, it has to be actively uptaken through hexokinase. Once it does that, it gets into the cell. We then don't know what happens to that substrate ongoing further. So PET, that's why it's useful for viability, because if it's taken up into the cell, chances are the cell's alive, but it won't tell us what's going on in terms of downstream metabolic activity. You'd have to use other PET traces, which generally have to be generated through a cyclotron, such as acetate, urea, the various other traces, which not every center has access to. So that is one of the differences between pyruvate and this C13 technique and PET imaging. The other important thing to note too is that unlike PET, where you can take, say, fatty acids and use them to look at what's happening to fatty acid and beta oxidation, with this technique, to hyperpolarize anything that's a long carbon chain which exists in a fatty acid is really, really difficult. It's not very stable. So that's a work in progress. And at this point in time, we've not really gone down that route to see what's available. So we've got a question. Yep. Yeah, one question. Yeah.
Can all these things happen within 60 seconds?
Yes.
Well, okay.
Exactly. So what I'm going to show now is how we actually read it out. So this was so first was the process of actually being able to do DNP in the rapid dissolution. Next, we had to develop a sequence that we could then identify all the key components of the metabolic products that we generated. And this is where, and this is all work from Angus Lau, who's now a postdoc in at Oxford, who did his PhD here with Chuck Cunningham. And this is really all Chuck's work. So I'm presenting it on his behalf. But essentially what the previous sequences that we used were these chemical shift sequences. And one of the problems with them when you do an MR spectroscopic readout is you'd have to do about 100 excitations. Now clearly if we're trying to move towards in-man imaging, we need whole heart imaging ideally. It needs to be done within a reasonable time frame, let's say 20 seconds. So that means we need to completely modify the sequence. And ideally we'd want it to be 3D. So that means we can get a whole heart, breath-held, 3D image that we can reconstruct. The previous way in which this was predominantly done was with these CSI images, where you'd have to do about 100 spectral pulses, and then you'd do sort of these single shot case-based trajectory readouts. So what Angus came up with this was to do a dual band excitation pulse, and essentially what it enables to do was to do a negative pass band and a positive pass band within a very narrow window. And what that means is because bicarbonate, lactate, and pyruvate all resonate in a different frequency, if you send basically a narrow enough excitation, they will have different resonant frequencies, and we can pull out each of those components. So we can look at, in the same 20-second breath held, whole heart 3D volumetric readout, bicarbonate, lactate, and pyruvate. And this is what an image looks like. Hopefully it'll play in a heart pyruvate here. Did it work? No, for some reason that color's not working. Let's play it again. Let's see if that works. Okay, so this is not going to work, which is great. And essentially what we had done here, this is this multi-slice dynamic image. So instead of doing a spectroscopic readout, this is a pseudo-colorized image. And what I had done here is pyruvate, we can see here is this yellow-red signal. And unfortunately, I don't know why it's playing, but bicarbonate, we color-coded green and lactate blue. And what you could see as you do this timeout to 15 seconds was that, if I change that, no, unfortunately, it's not going to work. You could see that we were able to resolve each of those three components with a single C13. And we've done, obviously, the time course readouts. You need to do this, though, at around 20 seconds. After 20 seconds, you'd start to get rapid signal decay. This readout, though, takes about 20 seconds. So it's as soon as injected, we start acquiring. We can acquire multiple times. We let it run to about 30 seconds generally, but then we can resolve the pyruvate, bicarbonate, and lactate peaks there. And we can also undergo MR spectroscopy to demonstrate what we're seeing with the pseudo-colorized images correlate to what we see with the chemical shift that you would see with the standard spectroscopic readout. So we're fairly comfortable that we're actually measuring pyruvate, bicarbonate, and lactate there. So that's a lot of background. So let's just move forward things to where we're taking this technique. So this is some pig studies. So obviously once we developed that, we were happy with the sequence. We decided, well, what we really want to do is get this into humans. So part of Chuck's work, not only developing the sequence, but of course we had to generate a new coil because you have to have a received coil that is tuned to the to C13. So this is a dual-tuned coil, and you can see here's a pig here. This is in a GE 750 3T scanner. There's Jen Barry there, nice photo from behind. I'm sure she's happy with that. These are intubated and ventilated pigs that had undergone. This was a heart failure model. So this is that rapid heart failure model where we put a pacemaker in, we pace them. Ventricular pacing for 5 weeks at 180 beats per minute. We do turn the pacemaker off immediately prior to the MR scan, and then we turn it on. And we did, every week, we did an MRI study, which included P31 spectroscopy, the C1 and C2 labeled MRI, as well as standard cardiac function. So this is what it looks like. You can see here, this is just a still image. This is a baseline. This is at the 5-week time frame. Here's the susceptibility artifact from the pacing wire. So it was actually pretty minimal actually. And this is what happened over time. So you can see this is the little dots here demonstrate left ventricular ejection fraction, which fell over time. The volumes increased and the wall thickness got smaller. So you can see the heart's basically pumped less effectively. They started to thin out and there was ventricular dilatation. So our model does generate heart failure. Also by the 5-week time, the reason we stopped them was the animals started to look sick. They actually start to get heart failure. They start to have respiratory distress. They look unwell, and if we hadn't have sacrificed them, the vivarium would have forced us to. This is our P31 spectroscopic readout here, just to give you an idea of what happens to the phosphocreatine ratio. So you can see fairly early on it drops pretty dramatically. And if we go back to that Stefan Neubauer patients in humans with heart failure, we use that ratio about 1.6 as the key ratio. Early on, by approximately 1 to 2 weeks, we start to see a substantial reduction in that phosphocreatine ATP ratio. Drops dramatically by about 3 to 4 weeks and was really unrecordable by the time the animals were just about to be sacrificed. And this is what happened when we actually looked at the Krebs cycle dynamics. So this is a ratio of glutamate to pyruvate to give us an idea of what's happening. As I said, this is that C2 label. So Krebs cycle dynamics starts off pretty normal here, fell dramatically at 1 week, and remained persistently low. Now, this was actually not what we were expecting. We'd hypothesized that this would remain preserved until late in heart failure, and that's what the ex vivo and Langendorf preparations have told us. So this was an unexpected finding. And this is what, when we looked at the glucose metabolic findings, we actually anticipated that this would fall early and remain low, but again, you can see this was actually maintained quite late into the until the animals really developed quite advanced heart failure there. So this was not expected. Now this is really the first longitudinal in vivo study to be done looking at real-time both Krebs cycle and glucose metabolic dynamics there. And then we did a number of tissue sort of characterization to get a feel for where the key tissue-based markers were. You can see there are some of the key enzymes that are involved in the metabolic process, PDK4, phospho-AKT, PPAR-gamma, CPT1 were modified. The only caveat I'll put with this is when the animals were sacrificed. So it tells us what's happening at end. What we really want to get at is what's happening early on, because that's when we want to start to targeting therapeutic interventions for these animals. So interesting, but I think at least the tissue-based studies didn't really give us what we were going after. So what have we done looking in the diabetic setting? Well, this is actually a rat-based experiment. This is a model of type 2 diabetes called the Goto-Kakizaki rat. Now, this is a nice rat. It shares a susceptibility locus on chromosome 10 with humans. It's a spontaneous model of diabetes. It's non-obese and non-hypertensive. So it's actually not a bad model to study. It's a fairly mild model of diabetes. It takes about 50 to 60 weeks before it starts to develop much in the way of cardiac complications and renal complications, which we like because I think that more mirrors the human setting. It's a pain because they cost $400 each, and I've got to pay $1 a day to house them for 60 weeks. So these experiments get expensive. But what we decided to do there was, the other advantage we have with our particular technique is if we, depending on where we have the coil, we can deliver our substrate, and then we can look at metabolism at any different compartment we want. We can look at subcutaneous fat, liver, cardiac, the kidney vasculature, anywhere. It's just a matter of where we actually decide to send our slice selective excitation. So this was a slightly modified sequence with the more typical CSI type sequence, and this is what it looks like with an MRS spectroscopic readout. We can see our pyruvate peak as we just delivered that C13 in the first carbon position. So this will give us an insight into glucose metabolism. And you can see here's the metabolic byproducts, a big peak where you see lactate, suggesting that there is a substantial amount of LDH activity in lactate dehydrogenase. Very little bicarbonate peak, suggesting that pyruvate dehydrogenase activity is reduced. And this was in the heart. So when we looked at both the heart and kidney simultaneously, you can see that there were some differences in the heart versus the kidney, that the bicarbonate to pyruvate ratio was dramatically different in the heart as opposed to the kidney, whereas we saw similar trends in the ability to generate both lactate and their alanine to total carbon production. So it does appear that diabetes itself does generate different responses in different vascular beds. And the reason I think this is important is it tells us if we're going to start to target different therapies, you may have a different response in a different organ bed. Now, it may be that this is, we might want to increase pyruvate dehydrogenase activity in the kidney, but this may have a deleterious effect in the heart and vice versa, and we just don't really know at the moment. The reason we also picked the kidney here is it's an extremely potent metabolizer. Like the heart, it is a really highly active metabolic organ. We then went on and looked at why this may be, and I won't bore you with the details other than to say that one of the key features we noticed is that within this animal model, inflammation seems to be a substantial driver of the response we see, and we think that it directly reprograms metabolism through some of these inflammatory responses, which is mediated through the TLR4 technique. So we're working on that further at the moment. We've actually taken these animals and we're doing a number of intervention experiments at the moment. I can tell you that if you treat with some of the newer agents like the GLP-1 receptor agonists, which are these newer agents that are used to treat diabetes, again you see quite different responses in the heart and the kidney there. So that's really the work we've been doing. What have other groups been doing just in the last few minutes to finish up? Well, there is certainly an interest in using this technique alongside standard late gadolinium enhancement imaging to look for viability. Here's a porcine model with a 45-minute circumflex artery occlusion. And you can see here's the late gadolinium enhancement imaging technique, where there's really transmural involvement in the infralateral walls there. And along with that, you can see that there's a substantial reduction in alanine as a marker that this is not metabolically active in that area. And unfortunately, I've cut the image off, but we also see a concomitant reduction in bicarbonate imaging. So I think these techniques have a potential role to say, because at the moment when we do LGE, we're really looking for the presence of scar as opposed to truly viability. Maybe if we can image the two together, then we can see, okay, you've got scar. What about the surrounding myocardium? Is it alive or not? Will it respond to a revascularization procedure? And using these techniques, if it's metabolically active, we may be able to give more insight into that. The advantage of this, again, just to put it in context with PET, is that our spatial resolution is better. So we can get this down to about 5 by 5 by 8 millimeters. That's our voxel size. So it's nowhere near as good as LGE imaging in terms of its spatial resolution, but it is better than routine PET imaging. PET MR, well, that's a different sort of, that's different again with a better spatial resolution as well. But this technique has not really gone a lot further, mainly because at the moment we're still struggling a little bit with that spatial resolution. We think really to get that into the usable range where I think it'll be clinically useful, I think we need to be in that 2 by 2 by 3 by 3 millimeter range, and that's actually quite a lot of extra technically demanding work on that sequence to get us into that range. So there are a number of other traces, and this is just to give you an idea of where the sort of literature is going in the field. James mentioned before, obviously, we're a group here at Sunnybrook, and as I said, the lion's share of the work's been done with Chuck Cunningham, but also the UCSF group, as I mentioned before, the Oxford group are really instrumental in that. They were the ones who came up with the idea of labelling the C13 at the first or the second carbon position, and Gorman's group there's a Swedish group and a Danish group. So they're really the main 5 groups in the world who are at least looking at cardiac applications amongst other things. This is just an idea in the cancer literature. So people have been looking at hyperpolarized C13. This is a glioma in a mouse. And this is another marker known as hyperpolarized fumarate imaging, which is a marker of cell death or apoptosis/necrosis. So we think there's a couple of these other newer markers which may give us more insight into what's happening metabolically. So just to finish off, where are we in terms of clinical trials? Because obviously it's all great to talk about animal data, but I think unless we can get this in and show its utility in humans, it really is an expensive technique. It's got a lot of development. To make it user-friendly, we need to show that this is going to have application in human studies. So this is, I've got an image here. This is essentially what the spin lab looks like. So this is a Health Canada approved GMP, which stands for Good Manufacturing Product, which means we can generate a product that is essentially been approved to inject into humans. It's a little more complicated than that, though, in that to get our pyruvate to be able to put into this machine to hyperpolarize it, Health Canada were a little upset about this syringe that we were going to use. So what we actually have to do is have these pre-packaged at the facility in San Francisco, and then they send this pre-packaged, irradiated, approved product over that then we can load into our hyperpolarizer, undergo the hyperpolarization process, and then we'll be injecting it just through an IV into man. This is essentially what it looks like when you open up the machine, and it's all it's a self-contained sealed system. We're a little surprised at the stringency of Health Canada. As far as I know, nothing can live below about 5 degrees Kelvin. So when we get it down to 1.3 degrees Kelvin, it's a little unusual that they were so worried about the infection risk. I think there's a lot of other things that are more likely, but God bless them. So this is the process that's gone through. It has been Health Canada approved now, so we've got the go-ahead to do the first in man. And obviously the next question is, well, which patients do we scan first? So really the first group that we're going to do is obviously 6 healthy volunteers, just to show that that injection itself does not cause any unexpected side effects. But the next group we really want to focus on is those patients with cardiac hypertrophy. Now, why is this the group, those patients with left ventricular hypertrophy? Well, 1, they're a very stable group. 2, there's a lot of patients with left ventricular hypertrophy. 3, it's a very major predictor of the development of heart failure, myocardial infarction, cardiovascular mortality. So it's a well-recognized endpoint for major adverse cardiac events. And 3rd, there's a lot of controversy in the literature about how they actually undergo their metabolic profile. Do they predominantly rely on carbohydrates? Do they rely on fatty acid oxidation? And there's a bit of a difference between what we're seeing in vivo and in vitro. So we think this will be a very interesting question. And I think once we get more experience with the technique and more happy with that, then I think we can move on to sicker patients. But obviously, it's the old Latin term, first do no harm, so let's stick to a healthy group first. So overall, and just to conclude, I think C13 is a promising technique, but like everything, it has its pros and cons. So pros: no radiation, and we can do repeated injections. That's clearly a pro. We can distinguish between the injected tracer and the downstream metabolic products. The tracers are physiological compounds that should be safe. We've pretty much developed a rapid acquisition system in terms of our sequences, and the spin lab can be fairly easily incorporated within the existing MR facility, but you have to essentially place it very close to the magnet because you don't have a long time window to get the product before you inject it. So really, practically, it needs to be next to your magnet. So if your magnet's already in a certain position and there's no space, that is a major practical reality. There has been 40 patients have had it to look for prostate cancelling. There were no adverse reactions, so it does appear safe. But like anything, there are major disadvantages. The time window, the cost is expensive. So how do you cool things down rapidly to 1 degree Kelvin? Helium. Helium, as you know, has been delicensed under the Geneva Convention, which was set up when the US decided they were going to stockpile large reserves after the Second World War. Very expensive to maintain those facilities. The US have then removed them. So nowadays, helium, being an inert gas, diffuses through the atmosphere. So every time those people use them at the kids' parties and talk in a funny voice and breathe out, the helium diffuses throughout the atmosphere and is gone. So we used to spend about $5,000 a year for helium. Now we spend $250,000 a year. So we've got helium extractors so we can recycle the helium. So this is not something that's just going to be in next to every magnet. Clearly you need skilled equipment, people who know how to use the equipment, and personnel. Development of all this stuff's a problem, and I think everyone appreciates that with developing new MR techniques. The process of DNP, as I said, we're really stuck with acetate, ketone bodies, small substrates like pyruvate at present. Long-chain fatty acids, really going to be a problem. And the other thing which is important, we don't yet have the same sensitivity as PET. PET is exquisitely sensitive. So I certainly don't see this as really a major competitor for PET, but I think it's going to give us added useful information that is really likely to remain part of the research realm. I would be surprised if this is ever makes it into more widestream use because of the issues that we've seen. I think it's going to be useful for understanding mechanistically what's going on, but this is not going to be something that every magnet is going to come with a spin lab with it. So with that, I'll just stop and take time for questions. Sorry, I went a little bit over time, but obviously thank all those who did the work. So really, Chuck Cunningham, a brilliant guy who's really led the way for all this, along with Albert Chen from GE. Angus generated the sequence there, and Marie Schroeder, who's a postdoc who's back and forth, just taking a job in Denmark, and then the MR group here, Graham, Jen, Yiping, Julie Green, William, and Masoom Haider, who's been doing all the work to help with the first-in-man prostate studies, and then the funding bodies. So with that, I'll stop, and thank you for your attention.
Thanks very much, Kim. That was really a phenomenal talk, and I think really highlights some of the amazing work that's being done in Canada in this field. And I really want to congratulate your team on doing it.